Now we'll discuss the applications of natural biological self-assemblies at nanoscale. They are basically used in diagnostic and sensing activities. For example, different kind of optical sensors, self-assembled DNA nanostructures loaded with intercalate dyes for tracking and labeling the biomolecules like proteins. Similarly, we can form electrochemical sensors, for example, detection of phenolic compounds, peptide nanotubes at the surface of gold electrodes. These are also used for the gene and drug delivery of drug by viruses. Nanofilaments loaded with deoxyribonucleases penetrate breast cancer cells and they promote the apoptosis. For example, there are different systems that can deliver drugs to the specific sites. Sample methods include smart drugs, then is the nanocomposite hydrogel systems and the magnetic nanoparticles. In case of smart drugs, here is the antibody and toxin. We will make the immunotoxin. That includes the antibody and toxin. That basically, immunotoxin is a smart drug that attacks the specific antigens. Immunotoxins are the proteins in nature that consist of antibody part and the toxin part. First of all, you will produce or you will form a immunotoxin that will attach with the antibody part, attaches to the protein on the cell membrane, and then this immunotoxin enters the cell by endocytosis and toxin destroys the cell. As a result, their apoptosis occurs. Second drug delivery technique is the nanocomposite hydrogel systems. That is basically follows the thermotherapeutic process that releases drugs that are encapsulated on heating. Gold nanoshells and nanoparticles can be used. Ideal wavelength is between 800 to 1200 nanometers. Then we can use magnetic nanoparticles for drug delivery. For example, drugs are bound to the magnetic nanoparticles that carry drug to the malignant sites with magnetic field, and these release the drugs by the enzymatic activity. Similarly, nanoparticles or biological assemblies are also used for the disease detections. For example, cancer or virus detection can be performed using carbon nanotubes, gold nanoparticles, and nanodots and nanowires. Similarly, gene detection, for example, the nanovirus. For example, in case of cancer or virus detections, carbon nanotubes are used. These are covered with the monoclonal antibodies. These antibodies for the growth factor receptors commonly found in cancer cells and currently increases the measure. Similarly, silicon nanowires, these are used to form the nanotubes and antibodies attached to the wire, and current changes are measured, can be applied to cancer cells and viruses. Gold nanoparticles and nanodots, that are similar applications. These antibodies attach to the nanoparticles and then nanoparticles antibody bind to the cancer cells and color reflected by when light hits these particles. Shape and size affect the color. Similarly, you can use the detection of pollutants. DNA strands functionalized at the end of our FRET probes that are designed for the selective sensing of mercury and lead. Chemical storage, building nanostructures with porosity and gas adsorption properties could be used for the peptidic residues such as zerogel was used to absorb the iodine vapors. These are also used for the imaging techniques. For example, conventional techniques, they have the limitations that are difficult to track the movements, and molecular tracking using quantum dots are labeled, and these can easily detect any problem within the body. They can track the blood flow and they can track the cancer imaging. Thank you.